Okay, in this video we are going to be discussing the functional role and the biochemistry of pyruvate dehydrogenase. Now this is the enzyme complex that links glycolysis with the TCA or tricarboxylic acid cycle. So where we are going to start here is the final product of glycolysis. And the final product of that, of course, is pyruvate. Now, recall that glycolysis occurs in the cytoplasm. The pyruvate dehydrogenase enzyme, also abbreviated PDH, is located in the mitochondrial matrix. Now, that means that we need to get the pyruvate, which is found in the cytoplasm, into the mitochondria. Now the mitochondria is a highly regulated organ with a double membrane structure. So let me draw the mitochondria in a very large fashion here, and we will begin performing our initial reactions in the mitochondrial matrix. So what I've drawn here is essentially the inner mitochondrial membrane. The inner mitochondrial membrane is a highly regulated membrane that requires transport of materials from the cytoplasm into the matrix of the mitochondria. The outer membrane of the mitochondria is considered rather porous. Now there are not large gaps in the membrane, but there are pore-like structures. And these pore-like structures can allow the passage of molecules that are generally less than 6,000 molecular weight. Now, if we were to calculate the molecular weight of pyruvate, we would see that it is very, very, very much smaller than this cutoff size to pass the pore. So the passage of pyruvate into the inner mitochondrial space is unimpeded. This, of course, would be the outer mitochondrial membrane. Now, once we have our pyruvate in the inner membrane space, then we have to use a transporter to transport the pyruvate across the inner mitochondrial membrane. Passage of any molecule is highly regulated across this inner membrane. So the way we're going to transport this is that we are going to use the power of a gradient. So the pyruvate will bind to this transporter, and it will be co-transported with protons. This co-transporter is a symporter, and the name of it is pyruvate translocase. Now the fact that it's a co-transporter indicates that there may be a chemical gradient here that is driving this process to transport pyruvate into the mitochondrial matrix. Indeed, we have a chemical gradient of protons. So we have a very high concentration of protons in the inner membrane space, and we have a lower concentration of protons in the mitochondrial matrix. We will look at a later video how we generate this gradient, but suffice it to say for now that the proton gradient is established by the electron transport chain. And we'll look at these details in a later video. So this is considered secondary active transport. because it's the power of a gradient, chemical gradient, that has been established by other means using energy. We will see this energy at a later time, but it establishes a gradient that we are now using the power of this gradient to drive pyruvate into the matrix of the mitochondria. Now, once we have pyruvate in the matrix of the mitochondria, we have to process this pyruvate by pyruvate dehydrogenase enzyme in order to generate acetyl-CoA. So acetyl-CoA is going to be our final product of this particular reaction, and then we will have the substrate to enter the TCA cycle. So the first step to understand is that the pyruvate dehydrogenase enzyme is not a particular enzyme, it is a complex. So PDH is a very, very large complex of many, many subunits of enzymes. The primary enzymes are categorized as E1, E2, and E3. And each one of these mediators, complexes that form the complex, is represented in multiple copies. So the PDH complex are multiple copies of each one of these subunits. Now, each one of these subunits is going to perform a specific reaction with the pyruvate in order to process this to acetyl-CoA. So let's look at first the first enzyme in the complex, E1. This particular enzyme has bound to it a cofactor called TPP. TPP stands for thiamine pyrophosphate. This originates from a vitamin. So thiamine, recall, this is vitamin B1. So the first step in the reaction is that we are going to transfer a portion of the pyruvate molecule to be covalently bound to the thiamine pyrophosphate, the TPP. And in doing so, we will be generating a CO2. So notice that after we perform this reaction, we only have 2 carbons left on the TPP. That means one particular functional group of pyruvate, specifically the CO2, will be released. So this is classified as a decarboxylation reaction. Of course, the decarboxylation, the loss of the CO2, is what's driving the covalent link between TPP and the hydroxyethyl form of pyruvate. Now, the next step of this reaction is going to require the E2 enzyme subunit. E2 now has a group that is called lipoate. This is another cofactor. And lipoate is a rather a short chain hydrocarbon molecule. It has fatty acid structure to it, but most important of this is that we have 2 sulfur groups. And these sulfur groups are in the oxidized state. Now, the next step of this reaction is to transfer these 2 carbons from TTP onto the lipoate sulfur groups. This is actually a redox reaction because we are taking the oxidized form of the sulfur, which is the bonded form, and we are breaking this bond and reducing this cofactor on E2. So what our group will look like then is this. So notice what we've done to this molecule. We have oxidized the alcohol-bound carbon and placed this hydrogen with these electrons on the sulfur group. This will oxidize this particular carbon, creating a carbonyl, and then we are transferring the bond from TPP onto the sulfur group of the lipoate cofactor. Now the next step of this reaction that also occurs in the E2 complex, the E2 subunit, is displacement of this acetyl group with another cofactor. This particular cofactor now is called coenzyme A. The coenzyme A is written as such to show the important functional group in coenzyme A is a free sulfur group. This sulfur group is what's going to displace the acetyl group from the lipoate. And so when this comes into this reaction, we are going to generate this molecule, which gets released. This is acetyl-CoA. So this is the final product of the pyruvate dehydrogenase enzyme. We've transformed the pyruvate into acetyl, 2 carbons with a carbonyl, CoA. The cofactor is bound covalently. Now, what happens to the lipoate here is we have our sulfur group that has lost the acetyl group, and now we have a fully reduced lipoate. So the reducing power, the reducing equivalent, has been transferred in this reaction from pyruvate onto lipoate. And in doing this particular reaction, we are controlling these electrons, which we are going to be using in a second, and we are generating a decarboxylation reaction to remove this, generating the energy for this process, and we are attaching a coenzyme A onto this to generate acetyl-CoA. Now, in order to regenerate the enzyme complex, we need to return the fully reduced lipoate into its oxidized form to create lipoate ready to continue the performing reactions with additional pyruvate. This process will be done by the subunit E3. So again, we are going to oxidize this reduced molecule, and therefore we need to reduce something. And in the process, we are going to reduce another cofactor, which is FAD, again, flavin adenine dinucleotide, and we will transfer those electrons onto FAD to generate FADH2. This is a redox reaction that occurs in the subunit E3. And in doing so, by transferring these electrons to FAD, we regenerate the oxidized form of lipoate. Now, FAD is another cofactor that is permanently bound 2E3. Being a flavin molecule, this is another vitamin generated from the vitamin riboflavin. Now, in order to regenerate FAD, to continue the oxidation of these reduced lipoates, we need to oxidize FADH2 back into its oxidized form. And we're going to do this reaction by performing another redox reaction. And in this case, we are going to transfer these electrons to NAD to make NADH. Now, once these electrons are transferred here, these electrons will be put into the electron transport chain, and they will contribute to the generation of ATP. In doing so, we've regenerated the oxidized form of FAD, so we can continue the cycle for the pyruvate dehydrogenase enzyme. So this total reaction includes a decarboxylation, and it also performs a redox reaction. So this is an oxidative decarboxylation reaction performed by the enzyme PDH. One final note is to look at the standard free energy change of this enzyme complex, the delta G 0 prime, and it equals negative 33.4 kilojoules per mole. This is a huge negative delta G, so this indicates that this reaction is only one way. So the generation of acetyl-CoA from pyruvate cannot be reversed. It's a one-way reaction with this huge standard free energy.